Right out to Audrey on that one.
Ah, okay.
It seemed appropriate for the department.
Should I turn my mic?
Exactly, same better to use the audio and
Okay.
microphones.
Okay. No video, no.
So that's why I asked. I'm like, what is happening right now? I feel like my perception of what next week will be like is very wrong. I think I need to text Nick and be like, are you okay?
What?
Yeah. I have to check in really quick. Friday is not Friday. I don't know. I'm gonna go up early. I'm gonna be there for Peter's day, Peter's morning experience, and I apparently have to be like the AP's twice now, so I will probably get there.
Yeah, so we're actually looking into if we can find the
If you wanna come early, let me know. If you don't, that's fine. I wasn't planning on it, but now I am, and I also don't have the schedule for that day, so I was like, when should I be there? He's like, yeah, it's like during the morning. Oh. Until the conference itself starts at. I always work at Tuesday starts at 11. So I will be there.
Yeah.
I'll be there. Year's Day is starting at 11.
Right.
You might have like a standard package. We had journals up in there once for each new week, and the journal which goes to the spot. I think that's pulling it. It was the last time we did that in here. I can Are you going to conversation? Okay. So my perception is not that wrong. Like what I understood is that everyone was there, but I don't know. It keeps moving. Oh. That's not surprising. Thank you. I don't know. I told everyone, I mean, and I don't know if I'm making a big deal, but I told everyone I should go to people that are just there. They're like,
Hi everyone, thank you for being here. I'm going to introduce Dr. Pividori. He's an assistant professor here at the Department of Biomedical Informatics. As a biomedical informatician, his lab aims to advance precision medicine by developing a comprehensive multi-omics approach to understanding the complex interplay between genetics and disease. For this, his lab develops new methodologies to consolidate large and heterogeneous sources of biomedical information to extract biological insight to improve human health. Thank you for being here.
Thank you, Natalie. Thank you, everyone. So today, the title is Beyond Linear-Only Patterns in Gene Expression Data Analysis, but it's really about any type of data. So in our lab, as Natalie said, we work on the development of advanced computational methods applied to human disease with a systems biology approach. Hopefully, that will help us to build a multi-omics approach with precision medicine. So what we do, as anyone here probably for sure, is to try to get correlations, understand our data. So usually we have this tabular format where we have a set of objects with different measurements across different sets of features. And we are usually interested in computing this correlation matrix that can reveal some important patterns between these features. So the presentation today will focus on CCC, is the cluster match correlation coefficient, a method that we developed that can, we say it's a not only linear correlation coefficient. So why another correlation coefficient? That's the first question. So the most popular methods like Pearson or Spearman are simple and very fast. That's good, but they can only capture linear relationships in data or monotonic relationships. There are some recent developments like the distance correlation or the maximal deformation coefficient that can capture more general patterns, but they are very slow. And in addition, they cannot handle a mix of different data types, like categorical data, for example. And for us, this topic is important because in the lab we work with gene modules or groups of genes that have similar co-expression patterns. For example, here you can see two gene modules. Gene module 1 is a group of genes expressed in the heart that has a similar expression pattern. We have another gene module expressed in macrophages, for example. So we capture those gene modules and integrate them with genetic studies. And we want to understand if this approach can help us better understand the molecular basis of complex traits. So the way we extract these gene modules is basically using an unsupervised learning method. We've been using lately FLIR, which is basically a matrix factorization approach, very similar to PCA, but it has the advantage of incorporating prior knowledge in the training stage. So we can align these modules or LVs with this prior information. So gene modules are key in this approach. This is an example of matrix Z. It's an output of FLIR. So you can see that we have two modules, and gene A and C in this case are grouped together in module 1 because they have this high membership degree. So this is an example of FLIR output, but this is a matrix factorization approach, so it should capture linear only patterns like the one that you can see here, strong linear correlation. But if we have something more complicated, like in this real example that has a sort of mix of linear and no relationship between these two genes, we probably don't capture that kind of pattern with these kinds of approaches. I will show you later that this gene pair is explained by sex differences in expression. So what we want is a more general approach to capture, don't miss any relationship. There could be gene-gene relationships that are important to explain sex differences in health and disease, so we don't want to miss anything. We want to capture all these general patterns. So that's the goal of CCC also. So how do these coefficients behave in different data patterns? For example, this is the Anscombe's quartet. It's composed by 4 data sets described by 2 variables with exactly the same statistics, but different patterns. For example, this is a noisy linear relationship. This is a sort of subsampled quadratic relationship. This is a linear plus outlier, and this is nothing plus outlier. So you can see, for example, the Pearson correlation is exactly the same in the 4, although they represent very different patterns. That's because the mean and standard deviation is exactly the same. But CCC seems to be closer to the expected values here. So the way CCC works is to take all the objects in the data and cluster them using the 2 features separately, and then compare the clusters. And that's the similarity value. So for example, in Anscombe 1 on the left, you can see that the cluster, one of the clusters, the 2 clusters using the X variable match perfectly the clusters obtained from the Y variable. We generated a few more synthetic data sets. For example, this is a random independent data set where none of the coefficients find anything interesting as expected. Here you can see that CCC used 2 clusters when using feature Y and 5 clusters when using feature X, but it was not enough to capture a high value. We also simulated other patterns found in other real data sets, like this non-coexistent pattern, where all coefficients are significant, but in the quadratic and 2 lines one, only CCC is able in this case to find a significant and high value. So this is synthetic data. Oh, it's only 8 minutes. I will finish very early. So perfect. We'll have more time for questions. So if we mix the data types, for example, here we simulated 2 categorical variables. Categorical variable W has 2 potential values, blue or orange, and Z has A, B, or C, which are categorical values. So in the first example, there is no relationship between these 2, whereas in the second one, there is a sort of overrepresentation of orange values in the B values. Right? But with CCC, you can also mix categorical with numerical variables, because everything is at some point represented as a cluster, so we can mix these 2 data types. And in this case, we also simulated a no relationship and a relationship on the right. So the next step was to use GTEx V8 in whole blood. We took the 5,000 most variable genes across 800 samples and applied these 3 coefficients. This is the distribution of the correlation values across all the possible gene pairs. You can see that the CCC distribution is more skewed, with values closer to 0, with a median value of around 0.10, I believe. So next, what we did was to take the top 30% of gene pairs, so those prioritized by these 3 coefficients, and the bottom 30%, so those where the coefficients say there's nothing here. That's the simplification that we use. So we generated the results are here in this subset plot. For example, in this column, you can see that in 42% of the gene pairs, the 3 coefficients agree that there is a strong correlation in the data, so very likely a strong linear correlation. In this other case, the 3 coefficients agree that there is nothing, which is 34%. So an interesting point here is that in more than 90% of gene pairs, all the coefficients agreed, but we were more interested in the disagreements. So for example, here in this 21,000 gene pairs, CCC found a strong correlation. Pearson says there is nothing, and Sperman says neither high nor low. So we focus on this. I'll show you some gene pairs that we found. For example, these are 3 different gene pairs with these nonlinear patterns. Sorry, the triangles indicate the high or low for the 3 coefficients, and the colors indicate that. So these patterns were missed by Pearson and Sperman. That does not mean that they are not significant. Sometimes they are significant. But if you're building a gene co-expression network, for example, the values are very small, and you will very likely not consider the edge between the 2 genes. So that's what we mean by missed. So we used independent data sets, in this case, Giant, to try to replicate the pairs that we were not able to explain with GTEx metadata. For example, this pair was found to be part, or predicted to be part of the same biological process by Giant, and expressing leukocytes, which was consistent with the original source tissue. For other gene pairs, we found that the explanation was sex differences in expression. This is an example where the 3 coefficients are significant, but in relative terms, here the CCC values are way higher than Pearson and Sperman. CCC was able to, this pattern is present across all tissues in GTEx. CCC was able to capture all of them. However, if you look at some tissues, Pearson and Sperman, for instance, in this case, they switch the sign, although the pattern is exactly the same. And for other examples, the relationship was not significant anymore. So this highlighted for us the importance of considering sex as a biological variable. And also, there's a possibility to apply CCC to other data sets with a mix of data types. For example, in this study, we focus on gene expression, which is only numerical. But in other scenario, we could have a mix of these data types and probably could apply CCC. I would be very interested if you want to apply. So another thing, a plot that I forgot to include is that CCC is faster than MIC or distance correlation, but way slower than, of course, than Pearson and Sperman. So how you done here is a new research assistant in the lab. He has a lot of experience with CUDA and GPU, so he'll be working on adding GPU support for CCC. So if you want to give it a try, you can install it. We have a Python package. You can go to the GitHub repository. We are very happy to help if you want to use it. Also, I want to say thanks to the lab and our funders. So thank you very much for listening. Yes. So it was 15 minutes, but I'd love to hear about, I don't know, what do you think about this? Is it useful to try to find this relationship? Yes.
I think it's definitely useful, but like, I just, I was trying to get my head around like doing it this way versus like a linear regression model. And, you know, like you said, post hoc, you're like, oh, sex is, you know, important, but, you know, we would often kind of think sex could be important and put it in a model and kind of have that. But then I was thinking this has the advantage, like, you know, you could have those sorts of things like sex that you didn't measure, you know, that are causing a pattern like that and be able to find stuff. But yeah, I'm just trying to, if you have thoughts on this versus.
No, that's great. That's the kind of stuff that I wanted to hear. So yeah, it's true that, for example, one of the gene pairs, we tried to explain it with GTS metadata, but it was not possible. So something else is going on. So it's true that sometimes we are not measuring the cost of these differences. And it's also true that, I mean, linear regression model is very powerful. You can incorporate covariates. We can stratify by sex. So this would not be necessary unless there is something else beyond sex. So yeah, this is it's a great point. I think.
What is the dependence of CCC on sample size?
We didn't explore that too much, but I can show you an example here. For example, this was, yeah, for example, here, we have Anscombe 2, right, which is a sort of subsample quadratic relationship. This is not significant. The other two are significant. So these are only 11 points. So we didn't conduct a study to assess that, but it's definitely impacted.
I think it'll be good to study the dependence on both sample size and also the impact of independence between samples. Because there is likely to be a correlated structure across samples, depending on where these samples are taken from. For example, if you had 10 samples, but 5 of them came from patient 1 and 5 from patient 2, they are not totally independent. So you want to take that correlation structure.
I'm not sure if I follow. So you meant the independence of samples?
Samples.
So
why are you measuring the correlation between genes? Yes. I'm sorry?
What the dependency have to do with the correlation? I don't get that one.
Because the assumption here is that, for example, this all 11 points are independent of each other.
No, they are not making, I don't think they are making that assumption. The correlation is just a correlation. It's asking how they are related to each other, not dependency. We never check dependency on correlation.
I think what Arjun means is if you have, for example, repeated measurements of the same patients, where you use a hierarchical model or something like that.
That's right.
Yeah, that will be great.
Because
we don't include that here.
Yeah, for example, if you do measure Bayesian correlation and then you get a correlation of, let's say, 0.9, it means completely different things if the 11 points were observed independently or whether they were all dependent on each other. Because the way to assess that is to measure the correlation between all pairs of genes and look at the distribution and see where this observed value for a given pair of genes falls.
It would be interesting, for example, to have this sort of prior clustering of
Right.
samples and then sort of adjust the value using that. Something like that could be interesting.
Have you looked at multi-factor analysis for looking at the categorical and continuous variables together?
No.
That's something I've done for more exploratory analysis, and it could possibly help with that.
Yeah, absolutely. So there is a coefficient, the PPS score, something like that I read, that could also generate these correlations. But one drawback that it has is that when you compute a correlation between two numerical variables, it's one type of similarity value. If you mix things, it's another type. So when you put all together in a correlation matrix, that has an effect if you want to cluster or something. So it's not really a homogeneous or consistent value. So I'd love to take a look at this and see how it goes.
There's a question in the chat.
Oh, sorry. Special network calculated from Pearson correlation can be converted to regular network by incorporating additional information. Is it also possible to convert CCC to regulatory relationships? So I don't know what additional information means, but I don't see why not.
Could you repeat the question?
Could you read the question?
I did, sorry. Is co-expression network calculated from Pearson correlation can be converted to regulatory networks by incorporating additional information. Is it also possible to convert CCC to regulatory relationships? From a and another follow-up is additional information typically is transcription factor target relationship. So I still don't see why not. I don't know if someone else is interpreting the question better and wants to add something as well.
So I think that you can add, you told that perhaps you can add categorical variables like sex, perhaps you can add like the transcription factor interaction here. I don't know if it's like the question goes to here.
Yeah, I
but
don't know.
If you can add like the transcription factor interaction, like a binary matrix or whatever, perhaps you can just clusterize better.
Yeah, something that we were thinking about is to compare, I don't know if this is what you mean, but compare sort of a multi-dimensional variable instead of just a single dimensional one. And we can perfectly run a clustering algorithm on multiple variables, so that could be a possibility.
Yeah, I think that was my interpretation. Like if you could introduce a partial correlation to identify a regulatory network. So like if you adjust for another gene, for example, does that explain the relationship that you're seeing between two genes? That's interesting.
I think the sample set which you point out is really important over here. How many features do we have to have in order to use your model? Plus, how you are going to validate it? I mean, comparing it to different methods and say your performance is much better than the other model.
Well, we could generate synthetic data, simulate data, expect a significant relationship, mix linear, nonlinear patterns, use independent expression data sets like Giant to replicate gene pairs. Those are things.
As you said, you are replicating it, but how do you know the strength of your model versus whatever number you are giving me is true? That's what I'm asking.
So this is a question about calibration or something like that?
Yeah.
Yeah, our model outputs the adjusted run index of the clustering. So that's, I mean, it's a degree of matching between the clusters. So I guess we should work on an explanation of how to interpret that number. It just means
Yeah.
it matches more if it is closer to 1. It's independent if it is closer to 0. So that's our current output.
How do you figure out the number of clusters based on interval?
Sorry?
How do you figure out the number of clusters?
That's a great question. One of the parameter of the method is the number of clusters.
Oh.
So by default is 10. So we found that across different sample size it was enough. But if the number is lower, if it is 2, for example, it will only capture linear patterns. If it is larger than that, it can capture more complex relationships.
Then based on that, you might be able to work out something like the required number of samples, right? Because if you say 10 is optimal, then you need certain number of points within each grid to sort of get some account of a stable, robust measure.
Yeah, the
that
method
also tells you now the sample size.
Yeah, the method automatically goes from 2, which is the minimum, to the square of the sample size. And it uses percentiles to cluster the data. And we remove any cluster that is a singleton. So we don't consider those. So yeah, there is definitely some relationship between parameter and sample size.
It's just a thought, getting back to the idea of validating with simulations, like kind of the examples you showed were ones where there's something underlying like sex that, so I mean, you could simulate something like that, but then your SCOMB2 there's just like the other one is just finding nonlinear relationships. Like, I feel like we're often trying to apply one metric to a bunch of comparisons where some might be linear, some might be quadratic, some might, and like, it could be a benefit of this that you have one algorithm that can find relationships with different underlying distributions. So like, it could be cool to simulate data where you generate a whole bunch of different types of nonlinear relationships and then evaluate whether this tool finds all of them well or something that would.
Yeah, there are definitely simulators to do that. When we did this, we perform a more simple approach here. But yeah, that would be great to do a more comprehensive simulation study. One drawback of these methods also is the interpretation part. Because of course, I mean, you find something that is not linear, but what is it? Then you have to understand. Thank you so much.
Is CCC a metric?
That question is answered if the adjusted run index is a metric, then CCC is a metric.
I'm sorry?
If the adjusted ARI is the adjusted run index,
yeah,
I think it is a metric. I mean, you mean it meets these 3 properties.
Exactly.
I think the adjusted run index is a metric. Yes, it's an adjusted method for chance used in clustering. So basically, the CCC is that. Okay, thank you so much.
Thank you. That was really funny.
Oh, yes.
That's what I wanted to ask. And I only have to talk for 15 minutes on like a 3-hour meeting, and he decided that was the most time he did. And I got one question after I talked to him, it was really professional. I'm glad that I said really scientific. Glad you took away the most important piece of what I had to say. Yeah, but I need to address that really quick. So I told Thor that they like this love Thor thing that he's helping. So can't be outside of the room. He's on the room. And then I'll door trying to get to me on the other side. I need to like he'll go in his bed if I tell him, but there's like this 159.
For our next speaker, we're gonna have Dr. Neil. He studied biology and music in college, followed by a master's degree in anthropological sciences. After several years playing music, he returned to science, first managing the lab of Joanna Mountain before spending several years as an ancestry scientist at 23andMe. He attended graduate school at UCSF, working with Esteban Fischer and Jeff Wall. He was a postdoc with Carlos Bustamante at Stanford prior to joining the faculty at CU. He's deeply interested in questions bridging population genetics and epidemiology, improving our understanding of complex traits through statistical genetics, biobanks, and big data, particularly across diverse populations. In addition to being in DBMI, he is the MPI of the NHGRI and NHLBI sponsored cage study and the director of research for the Colorado Center for Swine Medicine. Thank you for being here.
Thanks so much, everyone. Wanted to talk about just a couple of ideas that have been sort of floating around in the lab and with some of our collaborators and how we think about science. So yeah, of course, you know, we're here in this building. Our lab tries to understand kind of, like Natalie was saying, this intersection of kind of who we are and how who we are impacts our risk of various complex diseases in particular. So trying to do that at scale and in diverse settings and stuff has been a huge focus of the past 15 or 20 years or so. But really, you know, as a way of introducing myself, because people have asked in the past, here's some, you know, ubiquitous guitar playing figures from when I was very broke. So it's nice to get back into science and have an opportunity to work at a place like 23andMe when there were fewer than 10 people there, which was really eye-opening and really great. And just kind of to where we are now, in terms of thinking about what science actually means. There's a lot of different aspects of science for a lot of different people. And credit to Audrey on this one for using some GPUs to make this, about just how much of the job and trying to harness the best data sets that we can to answer questions, whether they be specifically focused on genetics or leveraging genetics, omics, and other kinds of things. That really a huge part of the job ends up being herding cats. It's definitely something that we've gotten comfortable with as we're trying to, you know, assemble the right data sets and be able to ask the right questions. I'll talk about a couple of different aspects of this. One of the things that I wanted to talk about today, what's up, Nick? Is, you know, why do we have this focus on ancestry as we're thinking about this kind of thing? And you know, ancestry means a lot of different things in a lot of different contexts. And, you know, certainly there's aspects of it that are relevant to genetics and parts that are relevant not to genetics. But one of the things that we really need to highlight, and hopefully if folks saw the announcement, there's an evolutionary genetics speaker who's invited to HMGGP next hour, who will be really great for those who can attend. But really, when we think about human genetic history, it's incredibly long and complicated for a species that like we're fairly young in the grand scheme of sort of evolutionary history compared to a lot of other species. People have moved around quite a bit. And of course, you know, sort of thinking about these early migrations doesn't really explain the entire story. What really does is the fact that there's been an enormous amount of demographic complexity in our species. And, you know, this is just sort of, you know, we can overlay as many arrows as we think migrations have occurred. And certainly if we think about what's happened, you know, in the past 500 years in particular with large scale immigration, both voluntary and involuntary. And all of this gives rise to the patterns of genetic variability that we see in modern populations today. So all of us in this room, on Zoom, even Nick on the couch, carries, you know, this sort of genetic history, right? This genetic legacy of all of our ancestors and where they came from. And yet, you know, these kind of figures are now kind of ubiquitous in human genetics. But it really speaks to the fact that, like, as we sort of start to think about the kinds of analyses that people do, we keep studying the same people over and over again. And, you know, I think now people have sort of understood, oh, this is a huge problem in human genetics. I would argue that it's not a problem that's specific to human genetics. It's just because there's this constant like intertwine between ancestry and any of the statistical tests that we want to run. Geneticists are sort of forced to account for this on a regular basis. And in a lot of other biomedical research, people just don't, or haven't been sort of forced to quite as much. Although we're seeing this now in things like clinical trials where the FDA has made improving representation in clinical trials a large priority. And so, you know, we have to kind of be mindful of this and kind of recognize the fact that, like, you know, people are pointing out that this is a concern in the field, but at the same time, as initiatives get larger, particularly in places like Europe where there's nationalized healthcare, you know, the sort of knowledge gap probably continues to increase. And so, like, we've been thinking about this, and a lot of our other colleagues have been thinking about this a lot. This is sort of stemming into some work that we've been doing with our close colleagues over at Davis. And it's this question of like, well, you know, what can we do to either close the gap or kind of focus on scenarios where we can address aspects of complex trait genetics in non-European descent populations. So does this gap really matter? I probably don't have to convince people in this room that the knowledge gap of like non-white people in biomedical research is kind of a problem. But there's a bunch of reasons why this inequity persists. And thankfully, some of the initial reasons kind of have gone away. Like initially, a bunch of the arrays that people were using in genetic epidemiology were specifically ascertained on European ancestry individuals. So they worked best in European ancestry individuals, which caused more, you know, it sort of ended up with this kind of runaway snowball kind of effect. So, you know, of course, here in the US, people are particularly interested in funding research that specifically reflects what's going on in populations of the Americas. People always focus on sample size. And, you know, in a situation where there's minority populations, they tend to have smaller sample sizes. And if people are only thinking about statistical power, that can be a challenge. Oftentimes, and this is something that a lot of people have pointed out in the health disparities literature, there's this perception that other populations are compared against European ancestry or white non-Hispanic individuals as the normal or baseline or benchmark or something like that. I mean, it's, you know, normal or comparative to whom. One thing that's happens a lot actually in review is this question of like, well, if you're looking in this one specific population setting, how applicable is that to everyone else? And that's not oftentimes a question that's asked of people when they're looking at majority populations in genetics. And one of the things I think that's been really encouraging to see is methods that work to address kind of challenges in populations of mixed ancestry. And so for people who aren't as familiar with human genetics, this concept of admixture is ancestry received from sort of maybe disparate parts of the world, different continents, or different populations in general recently. So we can think about that happening like in the Americas over the past 500 years, or across South Asia over the past 10,000 years. People talk about admixture processes with ancient hominids. The speaker next hour for HMGGP might talk about that kind of stuff. Anyway, it's this question of like, as soon as someone receives ancestry from a mixture of places from around the world, there's additional kind of statistical considerations and challenges, which used to kind of scare people off from doing this work. Or I've heard multiple times at big meetings, at ASHG kind of level meetings, like, oh, you can't do GWAS in admixed populations, which is completely untrue. I think Kendra would agree.
Yeah.
And so one of the things that we have to keep in mind is as we're starting to think about population stratification, and this is kind of an open problem that we're interested in working on with some folks in the future, is this question of what does it mean to adjust for ancestry? And this is kind of a common thing that gets done in association studies. You include some summary measure, some static measure of genetic ancestry, usually principal components analysis. And this gives you a way of sort of accounting for some kind of genomic background level of ancestry. But in fact, what that does is it tends to penalize variants that do have some degree of ancestry information or genetic distance between populations. And so in this kind of instance, you know, this may work on average. You look at some average kind of measure of your test statistic calibration and things look good. And things look good because you're averaging out this over and under correction of ancestry. And so we're interested in this because there's a lot of evidence, and I'll show some examples of this, where there's a lot of loci in the genome that are relevant to complex traits, but tend to be highly differentiated or more differentiated than they would be on average. And those loci are, you know, sort of interesting, and we want to kind of work on some statistical tests that sort of appropriately calibrate for that kind of information. Currently, these kind of models don't exist, and we're hoping to kind of do this in the future. I'm happy to talk offline about some approaches or, you know, in Q&A about how we're kind of thinking about this kind of approach. And, you know, we've been really interested in doing this, and kind of one of the things that we think is really important is, you know, it's oftentimes hard in human genetics to have like a ground truth or a gold standard to work on. And so Lemon from my group put together this mechanistic simulation for studying admixture and looking at traits that could be confounded or have some degree of ancestry correlation driven by either genetics or environment. And this is an art package that's available now. It's from a paper that we published a couple of years ago. And one of the things, so in contrast to previous work where people have said like, oh, working in admixed populations is difficult or is impossible or whatever, what we showed kind of starting from a very basic set of simulations is that in fact, admixed populations have greater power, greater statistical power in discovery, both for quantitative and dichotomous traits. And part of the reason for this is if you look across the entire GWAS catalog, this is taking a large number of traits from the GWAS catalog and just measuring FST or genetic distance between European and African populations. For a large number of traits, each sort of tick on this wheel is one specific trait. And we can see kind of this dashed line that represents kind of this genomic background kind of ancestry. But for a number of traits, there's a huge number of differentiated loci. And so we think that there's kind of a unique opportunity here to think a little bit more about what's going on in these highly differentiated loci and kind of do a better job of characterizing those specific effects and their roles in complex disease. So beyond just thinking about discovery, one of the other things that we can do and our lab is really interested in is calculating genetic risk or characterizing polygenic scores. One of the nice things here, again, is that if you're deriving or training your polygenic score model in an admixed population that has contributions from, in this case, 2 parental groups, that admixed population provides a better sort of training source. And so this, you know, sort of works against the typical trend of, you know, people looking in more homogeneous kind of settings. We're trying to remind people that there's additional, you know, even if you're only interested in statistical power, there's benefits for looking in this kind of setting. So this is definitely something that we're interested in sort of extending in the future, and we've got some active projects on extending apricot to some other kind of population structure scenarios. So I just wanted to talk a little bit as well about some of the work that's going on in the biobank. since it's, of course, down the hall from us over here. And it's been really nice to see since I joined here for it to start from having no data generated at all to now being one of the largest institutional biobanks in the country, which has been really gratifying to see, particularly how young we are in comparison to a lot of places that have been doing this for a couple of decades at this point. So we finally published our High-Over CCPM manuscript earlier this year that came out in the American Journal. And this is just an example of the kind of overall density of phenotypic information that you get from biobank scale data sets. And it's really nice to be able to look across traits, and Lemon from my group gave a really great talk at the American Society of Human Genetics meeting last year on some of the work looking across traits for a number of polygenic scores. But one of the things that we're thinking about is like, CCPM, like any other biobank that's just generally collecting samples from American cities, is going to have contributions from a number of individuals of varying ancestry from all over the world. So this is just, again, a principal components analysis if people aren't familiar with kind of the human genetics side of things. Oftentimes, people from a similar part of the world tend to cluster together, and the CCPM dots here in gray kind of reflect the fact that there's similar ancestry from CCPM individuals to different reference populations all over the world. So one of the things that we're interested in doing, of course, is trying to use this information to assess better patterns of risk overall. And one of the ways that we're interested in doing this is kind of measuring ancestry in a different kind of way. This is using a method called identity by descent, a concept called identity by descent, which is this question of not just necessarily having ancestry as something that's sort of, broadly shared across like entire kind of continents or that sort of thing, but actually asking the question of whether two people share a recent common ancestor, maybe in the past 5 generations, 10 generations, that sort of thing. And we can measure that kind of information at scale using some tools that we published, sort of allow us to think about this stuff kind of at biobank scale and data sets like CCPM and all of us in UK Biobank. And what that allows you to do then is kind of get a pairwise kind of genetic sharing kind of matrix. And that gives us an opportunity to identify clusters of individuals that are more likely to share relatives within a group than between groups, let's say. And so we did this originally, again, with one of our older biobank siblings, the Mount Sinai BioMe biobank, where they had detailed information on ancestry from a number of individuals in their study. And what we could show is that a number of individuals cluster into populations. And these populations aren't necessarily perfectly captured by like census or OMB categories. And in fact, we can identify populations that really find scale. So like, particularly given the sort of demographics in New York, we could identify a number of different populations of Caribbean origin, for example. And then one of the things that we can do in a data set like CCPM, again, now we don't have ancestry survey information yet, although it's something that we're hoping to work towards, but is to say that we can apply the same kind of thing in our setting. This is from the data from our American Journal paper. And we have a number of these different clusters, and then we can sort of measure overall patterns of genetic ancestry using those color codings for different regions of the world. And the important thing here to note is I recognize that this is kind of a bit of a busy slide, but the important thing is that some of these clusters look like they have similar patterns of overall genetic ancestry, but they cluster discretely from each other. And so we think that that kind of information can be important. And so here's just kind of a representation of this. And there's been critiques of sort of thinking about tools like UMAP in human genetics. But one of the things that we think is kind of really important here is to highlight the fact that, in this kind of instance, UMAP doesn't, necessarily reflect all this kind of structure. So here's a couple of populations that we see, or genetic ancestry clusters that are clustering with other individuals from a Hispanic Latino ethnicity. Here's some European ancestry clusters, again, kind of falling in similar spaces, but not exactly the same. And so, there's sort of a natural question there to ask of like, well, how does this information allow us to sort of monitor what's going on in these populations at scale? And the important thing to note here is that, this sort of measure of ancestry is telling us about some of the clustering or some of the structure in our data. But it doesn't necessarily mean that if we observe associations or specific traits that are enriched in certain groups, that that all of that's now driven by genetics, right? We're observing something about the overall population structure, but in fact, we expect that a lot of what's specifically enriched in one group for another is likely driven by environment. So here's just an example of this, again, from a nice slide from another member of the DBMI faculty here, kind of showing that, when we think about disease, we have to think about all of this stuff in context. So this means, a need for more cats. So, we could do this across a number of traits. And I certainly don't have time to go through like all of the associations that we've observed. But we're trying to ask the question of how are specific clusters enriched for specific phenotypes, adjusting for overall patterns of genetic ancestry. So we're trying not to be confounded by that. And we can see a large number of associations and these kinds of volcano plots. But really, the important thing is to start to dig into some of this work. And, it's for people who have looked at this before, this is a kind of PHOS plot, but now we're asking the question if this information, if certain traits are enriched in specific clusters. And in fact, in this specific cluster here, we can see that there's an enrichment of specific skin cancer kind of values over here on the left, as well as actinic keratosis, which is a precancerous state of skin cancer. And these are just, a few examples, of course. This is highlighting some skin phenotypes, but, we can do this with a number of other traits. And so we can look at this stuff across a number of these different communities. Now, these are all, are primarily individuals who self-identify as white, non-Hispanic, have similar genetic ancestry overall, but clearly in terms of their clusters, there's groups that are at higher risk and are at lower risk. And so this is unlikely to be stuff that's driven by like, large scale genetic differences between these groups. And so one of the things that we're really interested in doing is overlaying information. This is based on 3-digit zip codes, but now we have geocoded information for a number of our participants. And you can start to ask these questions like, oh, maybe one of these clusters in general lives at higher altitude and is exposed to more UV. I wish it was that simple. It still is a bit of a mystery as to what's going on here. I'll be honest with y'all, although if people have suggestions or that kind of thing to think about, it would be certainly welcome from the research team. So, I just wanted to say, and just kind of wrapping up here, this is just like a couple of the vignettes of the stuff that we've been thinking about. But really, what we want to sort of highlight is the fact that, as we're looking at complex traits and as we're understanding patterns of behavior in health systems, and of understanding something about the underlying genetic ancestry of all these participants is really important. This information becomes very useful in specific situations, both as to aid in discovery and for us to improve our statistical methodologies. And certainly doing this stuff in biobanks is really critical, which is why we tend to huddle together for warmth with other biobanks and work on projects in collaboration with folks at like Mount Sinai and UCLA and Vanderbilt quite a bit. And so there's a lot of opportunities for other things. There's a ton of work that we're doing in the polygenic score space. I don't have time for all of that, unfortunately. But just to say, you know, it's really great to be in such a collaborative department, and a lot of us have grants and work together and write papers and grants together and stuff. So I think we're all better off for it, and happy to take questions.
Hi. I have like a very basic question. Are all the
Yeah.
clusters roughly the same size?
No.
No, okay. I was wondering about
No. That's a good question. So, I mean, some of them get so small that it's hard to say anything like with any statistical
Okay, yeah.
assurance. So these are the communities that kind of have enough individuals where we
Okay.
feel comfortable saying something.
Making some of it.
But yeah, I mean, it's an important point, right? Like if people are really interested in some rare disease or something like that, you're only going to see differences in clusters that are large enough to capture that prevalence. Yeah.
Did you think about incorporating epigenetic data to understand better the effect of the environment into these population variants?
Yes. So the question for those on Zoom was about epigenetic variation. That would be fantastic to do. Historically, it's been so much more expensive than germline genotyping. But we worked on methylation some. We generated some methylation data around COVID susceptibility and severity that like Ian Koenigsberg and I worked on quite a bit early in the pandemic. We'd love to do more. It's very exciting now that Twist came out with a methylation library that allows you to generate like 4 million CPGs. So looking at that information would be great because that's getting pretty low cost. Like now it's competitive with genetics and it should capture more things. It's going to capture environment. It's going to capture the sort of current health status of each person. So yeah, if anyone has any money that they put towards methylation in the biobank. Yeah.
I'm gonna ask a question and then but is there something known about the difference between SNPs that are conserved across ancestries and SNPs that are not conserved in terms of biology?
Yeah, great question. So they tend, if they're on the extremes, either whether their allele frequencies look really similar or whether they look highly divergent, they tend to have been under selection at some point. Again, it's a great thing to sort of follow up with the speaker next hour.
Okay.
Thank you. But yeah, they tend to have sort of more unique evolutionary histories.
Cool, thanks.
Thanks, everyone.